Welcome to The Explainer. Today, we're diving into some absolutely fascinating new research that's changing the way we think about the progression of colorectal cancer.
So let's start with a really powerful question. What if the secret to understanding a disease like cancer isn't just about whether you have a so-called bad gene, but about how loudly or quietly that gene is speaking inside your cells? You know, it's not just about the musical notes in the score, but about the volume.
And all this groundbreaking work we're talking about today comes from the Systems Computational Biology Lab at Sastra University. They've done some brilliant genetic detective work, diving deep into our DNA to uncover the molecular clues that signal how this cancer actually gets worse over time. So, let's get into the central mystery that these researchers were really trying to solve. What's the hidden code that cancer follows as it becomes more and more aggressive?
Let's be clear. Colorectal cancer is a huge global health challenge. We're talking about over 930,000 deaths in just one recent year. And a big part of the problem is that survival rates just plummet as the cancer advances. This makes predicting the cancer's path, well, it makes it absolutely critical. Right now, doctors stage cancer based on what they can physically see. The size of the tumor, has it spread. But that's only what's happening on the surface. The real drivers, the things pushing it forward, are invisible. They're written in what's called gene expression, basically the molecular chatter inside the cells that's telling them what to do.
Okay, so to crack a genetic code that complex, you're going to need a map and some seriously powerful tools. Let's take a look at the very clever method they developed to sift through just mountains of genetic data.
They came up with this really elegant, step-by-step computational workflow. You can think of it like a battle plan for finding the needles in the haystack. How to take a ton of genetic data and find the few signals that truly matter.
Now, this might look a little complicated, but the idea behind it is actually pretty straightforward. They started with raw patient data from a massive public database. From there, they cleaned it all up, getting rid of the noise, and then ran it through multiple layers of analysis, all designed to pinpoint the exact genes whose activity changed as the cancer progressed from one stage to the next.
So after all that incredible digging, what did this genetic investigation actually turn up? Well, the clues they found are fascinating, and they tell a really clear, stage-by-stage story.
And here it is. This is what they found: distinct sets of what they call stage-salient genes. Think of these as unique molecular signatures that were either way more active or unusually quiet at each specific stage of the cancer. It's like every stage leaves its own unique genetic fingerprint.
This quote from their paper just perfectly nails the core concept. See, it's not just about having a bad gene. It's all about which genes are turned up or turned down, and critically, when. They found that in the early stages, the protector genes, the tumor suppressors, were being silenced. But in the late stages, the growth-promoting genes were getting amplified. Exactly. Think of it like a volume knob. In the early stages of the cancer, the volume on the protector genes gets turned way, way down. The cell basically loses its damage control. Then, in the late stages, the volume on the genes that fuel aggressive growth gets cranked all the way up, telling the cancer to grow and spread.
And this methodical search led them to their most critical discovery, a tiny set of genes that could act as a powerful predictor. This is the smoking gun, and it has real implications for patients.
Identifying these gene patterns is one thing, but the real holy grail here is prediction. Can you actually look at this molecular signature and get a sense of what's going to happen to a patient down the road?
It turns out you can. The analysis zeroed in on this specific trio of genes, HOT-AIR, GABRAD, and DKK1, which were most active in stage 4 cancer, and the expression levels of just these 3 genes turned out to be incredibly telling.
And right here, this is the payoff. These charts show patient survival over time. Just look at the dramatic split between the two lines. The red line represents patients with high expression of those 3 genes, and you can see they had a significantly lower chance of survival. Meanwhile, the blue line, those with low expression, had a much better prognosis. It's a crystal clear link between gene activity and patient outcome.
So, the crucial point here is that this isn't just some interesting academic finding. This 3-gene signature is a powerful tool. It could, one day, help doctors identify which patients are at the highest risk and might need more aggressive forms of treatment right from the get-go.
But, based on all these discoveries, the team didn't just stop at publishing a paper. They actually built something tangible to help other researchers and, hopefully, future doctors.
They developed a tool called COADRX. And what it does is it basically bundles all of their predictive models into a user-friendly experimental website.
So this tool takes all that complex research out of the lab and puts it into a form that could one day help in a clinical setting, turning these massive data sets into real, actionable risk assessments. It's just a fantastic example of research in action.
And that's really why this all matters. It just reinforces this idea that by listening to the volume of our genes, their expression levels, we get a much clearer, much more dynamic picture of a disease than we would by just looking at a static list of genes.
Which, of course, leaves us with a final thought. This research provides an incredible blueprint not just for cancer, but maybe for any disease that gets worse over time. It really makes you wonder, what other conditions could we finally decode if we just started listening more closely to what our genes are trying to tell us?